Hi, can you hear me? Okay. Um, Welcome to the second session. We're going to be switching gears a little bit. Did a deep dive in analytical chemistry and a little bit of toxicology, and now we're going to be talking about looking at approaches to sort of get on what we think is going on in terms of analysis of community water systems and domestic well households, and also trying to understand and do what Dr. Antell likes to call forensics on PFAS sources in terms of their threats to drinking water. So I'm going to just quickly introduce all our speakers at the beginning, and then we'll just kind of go straight through it. So we have 4 great speakers. First speaker will be Dr. Ned Antell, who is a recent graduate from the Department of Civil and Environmental Engineering here at Berkeley, working with Lisa Alvarez-Cohen and David Sedlak, and his dissertation has focused on developing lab-based statistical tools to look and assess PFAS source apportionment. And he's now a staff scientist at Remedy Scientific. It's a startup developing technologies to remediate PFAS in soil. And I would be remiss, and he finished his dissertation and had twins recently almost at the exact same time. So it's amazing that he's here, although a little sleep deprived. Our second speaker is Wendy Linck, who's a senior engineering geologist with the State Water Resources Control Board within the Division of Water Quality. And she's managing the State Water Board's response to the PFAS effort within the Division of Water Quality. She's also a liaison with the Division of Drinking Water on this issue. And she got her bachelor's degree in geology from Sacramento State University. And she's a registered geologist with the state of California and certified as a project manager professional by Project Management Institute. Our third speaker will be Dr. Clare Pace, who's a research scientist with us here at the Superfund research program, and she's leading our community engagement core as part of our water equity science shop. And in addition to her scientific work, she has collaborated deeply with our community partners, the Community Water Center, to develop the drinking water tool, which is an online interface for diverse users to learn about their drinking water sources, drinking water quality, and threats of contamination and access due to drought. And last but not least, our fourth speaker will be Hailey Shingler, who is with the Community Water Center as a Community Solutions Coordinator. She formerly worked as a groundwater quality analyst for the Department of Environmental Quality in the state of Idaho. She works with the CWCs as doing technical assistance work in pursuit of achieving safe drinking water for all Californians. And in her spare time, she spends a lot of time reading on the beach and collecting vinyl records. So without further ado, I want to welcome our first speaker, Ned Antell.
Cool, thanks for the intro. I'll never get used to the doctor thing or particularly enjoy it. So yeah, thanks for that intro. I'm going to be talking about source apportionment and PFAS. And I want to start out by talking about my childhood home. So here I am at 3 weeks old. Here's over there. And so about 5 years ago when I was starting, or 6 years ago if I'm being honest, I was hanging out with my parents and we were driving around and they pointed out this spot and they said, oh, hey, look, this place actually applied for Superfund status, but they didn't get it. And I was like, this place is 2 miles from your house and, you know, we're on well water. That's scary. And I looked it up and of course it was contaminated by PFAS. So I had them send me a sample and lo and behold, it has, you know, fairly alarming concentrations of PFAS. So it's safe to say I was raised on the stuff. So, you know, shortly after graduating, my dad is here helping me out with some stuff, and he says, you know, there's and he said, you know, there's actually this former firefighting training area over by our house. And so suddenly, coincidentally, my PhD dissertation is also my childhood story. But I don't think this is actually really a coincidence where all of us are inadvertently being exposed to PFAS. And so we need good forensic tools in order to be able to prioritize cleanup, limit exposure, and hold parties responsible. And this feels particularly applicable. There we go. For my 3-week-old daughter, now 10 weeks old, you can kind of tell we're related. As well as my son. I don't know where he came from. He doesn't look anything like us. Yeah, this feels super important to me now, that we want to create a world where this is not where everyone is being inadvertently exposed to these contaminants. And so David did a really great job of talking about how PFAS is really a lot more than PFOA and PFAS. There are greater than 12,000 PFAS out there, over 1 million, depending on how you define it. And we only measure about 100 of these. And the problem with forensics is that a lot of these compounds that we measure routinely are common to lots of sources of PFAS contamination. So the panel of PFAS that you're measuring may not be diagnostic to a unique source. So instead, what we need to be doing is focusing on these other 12,000 or more compounds for creating fingerprints that might be unique to different sources. And so when I'm thinking about forensic tools, I tend to think about it kind of in two different parts of developing the tool. There's first the data gathering, so how you actually measure PFAS, and then what you actually do with the PFAS data once you have it. And I want to say also that these tools are always going to be supported by conceptual site models and hydrogeological modeling. But for the purposes of this talk, I'm really just going to be talking about tools that rely on actual measurements of PFAS. So just to give a little bit of background of different options out there, first and foremost is obviously targeted methods like 1633. We've talked about that. Benefits of that is that these are standardized, defensible methods. They probably have the greatest data availability for those reasons. The downside is that the list of compounds, as Jean mentioned, 40 is not enough, right? And it's probably not enough for forensics either. At the other end of the spectrum, non-target analysis, this is an extremely inclusive method. You're measuring many, many, many different compounds. But as we've talked about, these methods are hard to standardize. They're semi-quantitative at best and require a lot of expertise. And so the top assay is kind of like this sweet spot between those two. There's moderate data availability. It's not yet standardized, but there's efforts. And it does a pretty good job at measuring a lot of different compounds. In terms of data analysis, similarly, there are a lot of different options out there. There's probably the most traditional method being positive matrix factorization. This method suffers, though, because it relies on this assumption that the composition of PFAS does not change as it moves through the subsurface. We know very well that this is almost never going to be the case, as there's preferential partitioning of different charged and different chain length compounds. Principal component analysis is a similarly traditional, pretty straightforward linear algebra method, but is a little bit more exploratory rather than quantitative. There's been some cool work on Bayesian inferencing using top assay data as inputs that has some really neat results that I'd encourage you to look at. And then, of course, machine learning tools have been garnering a lot more attention. You plug in some training data, black box, it spits out a classification of what it thinks what it thinks the source is. And so there have been a lot of different studies out there kind of mixing and matching all of these tools, and how you actually mix and match really affects both its efficacy and who it's available to. So I think there's a lot of thought that needs to go into how you actually design these tools in that regard. And the one that I'm going to be focusing on today, and which is the topic of my first chapter of my dissertation, is using top assay data to feed into a PCA model for source apportionment. And this is a pretty simple way of doing things. Not a lot of like glamour and hot button, or not hot button, kind of like, not a lot of like glamour behind this, but I think the simplicity is really what makes this desirable, because it's widely applicable and it's fairly easy to understand. Okay, so jumping into the methods. So as I said, there's pretty decent data availability out there. So rather than repeat a lot of effort, and while I was cooped up in my house during COVID, I just kind of scraped through the internet finding top assay data from peer-reviewed journals, public databases, researchers, and then some in-house samples that were actually provided by Wendy, so thanks for that. And then, so this resulted basically in 153 samples from airports, wastewater treatment plants, and landfills. You can imagine that this kind of looked like this, where each row is a sample or an observation, and then we have PFSAs, PFCAs, and then what I'll call delta PFCAs. These are the PFCAs that were yielded from the top assay. And so I want to just first kind of talk about what I'll call like fingerprints. So this is the mean bootstrapped relative abundance of the PFCAs, PFSAs, and delta PFCAs from the 3 different sites. And this kind of gives you a sense for what species are most abundant at these different sites, to give you a sense of, okay, this has this kind of fingerprint, this has another. So looking at airports first, well, we see that C8 PFSA, which PFOS, is really abundant. This makes sense because we know that PFOS was a legacy component of AFFF. Then we also see that C6 PFCAs are also very abundant. This is also unsurprising because the C6 precursors were the replacement products for PFOS in AFFF. Then moving over to wastewater treatment plants, you really see the importance of precursors here, where basically, I mean, the vast majority of the mass is all tied up in precursors, particularly in the short chain compounds. And then finally, going over to the landfill, we see this kind of unique signature with having high abundance of C4 compounds, particularly the PFCAs and delta PFCAs. So you can kind of see that with the addition of precursor data in these plots, you kind of develop a fingerprint that allows you to separate these out. And these patterns were also clear in the PCA, which I'm going to show now. This is showing PCA of the abundance data without including top assay data. Each color is the different source. So sorry, the legend will show up in the next part. The blue is airport samples, the orange is wastewater treatment plant samples, and the green is landfill samples. And I've drawn a 95% confidence ellipse around each of these sources. And what should be clear is that when you exclude top assay data, all these ellipses overlap. So the first 2 principal components are not able to separate out the different sources. If we then include top assay data, these ellipses separate out really nicely. You see that the area of overlap between airport samples and wastewater treatment plant samples is greatly reduced, and then the landfill samples are completely separated off. So this was exciting, but I think it's important that this type of work doesn't just stay within academia. So what we did is we converted this into an online tool where anyone with top assay data can log on to this website, download a template CSV, copy and paste their top assay data into this, upload it, and then a little red dot will show up on this plot. And to give you some sense of a potential source in your sample, and of course this only works if these 3 source types are your suspected sources. But we could build this out further with greater data availability. But it's important to note that all of the data collected in this, I'll call it training data, right, was collected directly at the source. And when you're doing source apportionment, by definition, you're going to be collecting your samples away from the source. Otherwise, it's not source apportionment. So when you do that, there's an opportunity for samples to be mixed, and that makes things more complicated and makes things harder. So what we did was kind of simulate what mixed samples would behave like, and then see how this model performs. And so we, simulated mixed samples at 3 different ratios by combining all these different samples that were included, and then plotted them, drew an ellipse around them, and then see how they move. And what should be apparent is that as you increase in the proportion of one source, it very predictably migrates from one ellipse to the other. And this is one of the strengths of PCA. It's based on linear algebra, so you have this linear behavior. Where it could get you into trouble is, so for example, on this leftmost plot, we're going from airport to landfill. A 50-50 mixture could end up looking like a wastewater treatment plant sample. And this is where we could add in a larger panel of analytes that could further separate these out. There's always opportunity to improve this. And then what I've alluded to also is that transport is really important. As a sample moves through, or as a plume moves through the environment, we really have no idea what it's going to encounter, how that's going to impact the fingerprint. And testing out the effect of transport is really difficult in the environment because we don't know what the true answer, or the true source is. So as a way around this, my lab mate, Katerina Zhao, ran a column experiment where she loaded a soil with AFFF and then eluted it over time, and then to kind of mimic transport through the subsurface, and then ran top assay on a ton of different samples. It was a Herculean effort. And this was a super encouraging result. We see basically almost all of the samples that are coming out of this AFFF contaminated column end up in the AFFF ellipse, regardless of soil type or pore volume or elution matrix. So this was super encouraging, and it also makes me think, okay, we should be doing this on other matrices as well. So contaminated, like for example, biosolids or landfill contaminated soil, and see how this could impact the fingerprinting. So as I've been mulling this over for several years, there have been a handful of questions that I don't have the answer to, but wanted to share with you all. The first being, is real-world validation of a forensic tool possible? Is it like, can we go out into the world and introduce or measure PFAS as it moves through the subsurface to validate these tools, or does it just get too complicated because we can't actually know what's going on in the subsurface and where different plumes might be interacting? The next is, what tools are legally defensible? A lot of the application of this is going to be in a courtroom. And I'm not a lawyer. I don't think anyone in here is a lawyer. So how do we make sure that what we're doing is actually useful? And so I think some sort of like cross-pollination could be really beneficial for our field. And kind of along the same lines, not just what's legally defensible, but like what is actually going to be understandable to a jury. If I start talking about like linear combinations and hyperplanes in a PCA, like you're going to glaze over. So how do we create tools that are also accessible, but still powerful and accurate? And so while we are an extremely litigious society, I would like to think that there are applications of forensics that are outside of the courtroom. And so thinking about ways to empower communities to also hold parties responsible that don't revolve around taking people to court. And then finally, all of these tools are always going to be improved with more data, but data sharing is complicated, and identifying who should be responsible for collecting all this data and maintaining this data, I think, is an open question. And then it's also just kind of it's a hot button issue. So how do we convince people that, okay, you can share data and sanitize it in some way and still protect your client or yourself? So yeah, that's kind of it. Thank you.